This video is a summary of cellular respiration and mitochondria, the events of glycolysis and oxidative phosphorylation inside a mitochondrion. Here's a mitochondrion, the outer membrane and now the inner membrane. The space in the inside is the matrix, and between the two membranes is the intermembrane space. I'm going to start with glycolysis. Before we even get to events in the mitochondrion, we start with a 6-carbon molecule, glucose, and I'm just showing the 6 carbons. In the first half of glycolysis, we require 2 ATP, 2-adenosine triphosphate, to break that 6-carbon molecule into 2 3-carbon molecules, each with a phosphate, PO4-2. In the second half of glycolysis, we take 2 NAD plus and 2 phosphate, along with 4 ADP, adenosine diphosphate, and we remove the 2 phosphates from the 2 3-carbon molecules, and we end up with 4 ATP, 2 NADH, and the 3-carbon molecules are converted to pyruvate or pyruvic acid. The two forms interchange. So we have a net of 2 ATP from glycolysis. The pyruvate is moved through pores. There are large pores in the outer membrane of the mitochondria, and then through a transporter into the matrix of the mitochondria. One of the first things that happens is a carboxyl is removed as a carbon dioxide, and then the 2-carbon piece remaining is added to a 4-carbon molecule to make a 6-carbon molecule. And then in a cycle, step by step, we go back to a 4-carbon molecule which can pick up a new 2-carbon molecule derived from a pyruvate. Along the way, 2 more carbon dioxides are removed, so that's the 3 carbons with the first one up above and then the other 2. And along the way, 3 NADH are made from NAD+, and 1 FADH is made from FAD. This is the citric acid cycle, part 1 of what goes on in a mitochondria. And it takes place inside the matrix. The second series of events happens between the matrix and the intermembrane space. This is electron transport, and there are a series of 4 protein complexes. They're big molecules, each made of multiple proteins, and a couple of interacting ones I won't name. The 4 complexes are involved in using NADH or FADH. The events are that from each NADH, 2 electrons are moved into complex 1 and then through the other complexes, and in the process, those movements of electrons cause changes in shape in each of the complexes, such that complex 1 is a proton pump. It moves proton across the membrane because of its response to the changes in shape from the electron. Complex 3 and complex 4 also move protons across the membrane. So they move protons out of the matrix into the intermembrane space. FADH joins at complex 2. So an FADH doesn't move as many H pluses across the membrane. And both FADH and NADH are regenerated into NAD plus and to FAD to continue the citric acid cycle. This is electron transport, and the end result of electron transport is to create what's called the proton motive force, a concentration and charge gradient of protons, and it functions as a proton pump. What happens to those electrons? Those electrons wind up being given to oxygen. So 4 electrons are given to molecular oxygen, forming O-2, and those 2 O-2 pick up 2 protons to become water. The third step is ATP synthesis, and I'm showing here the part of ATP synthase that is in the intermembrane space If we zoom in, the parts that you might be able to see are in green are two channels that allow a proton to move halfway in. The matrix is 200 millivolts, so there's a net attraction of charge. Hydrogens move through the little cylinder here. They're attracted to negatively charged amino acids within this cylinder. They move all the way around. The cylinder rotates because the hydrogens attract negative charge in the cylinder, and at the far end, hydrogen is pushed out because there's a nearby positive charge. Hydrogen has a strong tendency to enter because there's a 10-fold concentration gradient. It's pH 7 outside and pH 8 inside, so there's 10 times as many protons outside as inside, and it's negative, minus 200 millivolts. At this stage, the rotating cylinder causes the lower part that I'm drawing on now to rotate and push an ADP into contact with a PO4. This is an energy-intensive step because both of them have a -2 charge. It's hard to get them close enough to form that covalent bond. And the rotating of this whole molecule pushes the two together to get an ATP. So putting it together, first we have glycolysis. The first half, break a glucose into two 3-carbon molecules, each with a phosphate. That takes 2 ATP. And the second half, which we put in 2 NAD+, 4 ADP, and 2 phosphates, and we get back 4 ATPs for a net gain of 2 ATPs. So a glucose gets us 2 ATPs. If we don't have mitochondria, then it's just a fermentation reaction, and the NADH has to be regenerated. We give the 2 electrons and the hydrogen to pyruvic acid to form lactate or lactic acid, and then that becomes a waste product. In lots of organisms that undergo fermentation, there's other processing and alternative pathways. So other wastes are ethanol, for example, in alcohol yeasts, or acetic acid for vinegar yeasts. Finally, the steps within a mitochondria produce a net of about 30 or 32 ATPs. It depends a little on the reaction conditions, so there's no precise number that is universal.